it's a real honor for me to be here today and to be part of this homage to Ginés. If Akaimo said that he was terrified, imagine how I am after this line of speakers today, but well, I'll try to do my best. And yeah, I did my PhD with Ginés. It was almost 10 years ago, but if you compare that with Jordi, Akaimo, and Ernesto, that's recently. So I'm going to talk a little bit about science, but I hope that you also have an impression of how it is to work with Ginés and how influential it was for me to have him as a mentor. So one of the really remarkable things about Ginés, as you heard, is how many groundbreaking contributions he made in so many different fields. I think that's quite unique. And of course, there are many reasons for that, and he has many, many qualities that make him an extraordinary scientist. But there's one that I would like to point out today, and that's the fact that's how he reacts to unexpected results. That's something that was actually particularly important for me. And I'm going to illustrate that by telling you how he started working in apoptosis. Because, you know, all these years that he dedicated to investigating apoptosis and all the papers that he published in this field, and the fact that I think that now he is known as one of the leading figures in the field, all that started with one unexpected results. So first I'm going to, First I'm going to tell you the story, like the straightforward story, the story that you can read in the papers. And that is that we wanted to study signaling by apoptotic cells because apoptosis was supposed to be a silent process. Apoptotic cells were supposed not to affect neighboring cells at all. And then we found that this is not true, that apoptotic cells can produce mitogenic signals, and so they are going to induce proliferation in the neighboring cells. And this was found for the first time in Drosophila by Ginés's group and also by 2 other different groups. And later on, it was shown to happen in different organisms, and it was shown to be essential for different processes like regeneration and wound healing and tumor growth and relapse of tumors after treatment. And the way that we discovered this, or at least what I present when I give a talk, is that we had this very ingenious idea to keep apoptotic cells alive. So we did that because we induced apoptosis, but at the same time, we were expressing P35. And what happens then is that the cell keeps activating the apoptotic program all the time. The cell keeps thinking that they have to die, but they cannot die because caspases are inhibited by P35. So these cells become what we call undead cells. And this is basically, I always say that they are like zombie cells because they have all the features of the dying, but they are alive. So what we saw is that in this case, they were producing a lot of mitogenic signals, and this was producing overgrowths. So we found that indeed apoptotic cells are producing signals and are not silent. But of course, this is not how it happened. We were not interested in apoptosis, and we were not wondering about signaling by apoptotic cells. We were actually working on cell competition, that is this phenomenon that, as you heard today, Ginés and Pedro Ripoll discovered in 1975. And we were trying to find whether cell competition plays a role in maintaining size of organs. Because there's one curious aspect of cell competition, that is when you have one minute plus one wild-type clone growing in a heterozygous background for minute, that is slow dividing cells, what happens is that you get a very big clone that can occupy most of a compartment, but that compartment and the whole wing maintain the normal size. So it was hypothesized that this could be because of cell competition, because this big clone might be killing the neighboring slow dividing cells, and that's why the size is maintained, because if these slow dividing cells were maintained in the tissue, maybe you would have an overgrowth. So we were trying to test that, and in order to do that, we were generating these minute plus clones in the minute heterozygous background, but we were going to block apoptosis to prevent cell competition in the posterior compartment, and to do that, we were going to use P35 that was known to block apoptosis. And Paco was actually the one that was in charge of these experiments. I was only helping out here. I don't know if you know Paco. Probably you do. If not, you should. And so he was doing the appropriate chromosomes. He was generating the FRT chromosomes to do this experiment. But of course, this was going to take a while. And immediately, Ginés came saying, like, you know, when I was young, there were no FRTs, and you can induce mitotic recombination by irradiation. So why are you going to wait? You know, you can know the result in advance. And while you're doing the FRTs, you know what's going to happen. So aren't you curious about the result? So of course, we couldn't say no. So we started irradiating these discs. And then indeed, we were getting overgrowths in the posterior compartment of these discs. But immediately, we realized that this didn't depend on the presence of the minute clones or on the presence of the minute heterozygous background. As long as we had P35 there and we were irradiating these discs, we were getting these overgrowths. And so this was very weird. And I think that most PIs would have said, like, OK, throw this away, do the FRTs, and continue with your project. But Ginés was totally fascinated by this result. And he put me to dissect tons of these larvae, and I started producing tons of discs that looked more or less like this. And he would sit for hours and hours looking at these images. I think he saw every single confocal picture that I took of these discs. And so we didn't have a clue of what was happening here. But he was so excited, and he would be like, oh, I don't know what's happening here, but look at these discs. This is so exciting. This is incredible. We have to figure this out. So we started doing stainings to see what could be happening here, and we started talking about experiments that we could do. And in the end, we found out that what we were doing is we were generating these undead cells. We were generating these cells that had all the apoptotic markers that we could think of, but they were not dying. They were staying in the tissue, and they were producing high levels of mitogenic signals like wingless and DPP, which were responsible for the overgrowths. But of course, it was not only undead cells that were producing these signals. Normal apoptotic cells, we showed that they can also produce these signals, and we speculated, that is, as it was later on discovered, that this could have a role in regeneration or tumor growth. And so you can imagine, how exciting it was and how great it was for a PhD student facing a project like this, where everything was uncertain, to have a person like Ginés that was so exciting, and this was so contagious and so encouraging, and I was looking forward to producing more of these discs because it made him so happy. But it didn't only help me during my PhD. Actually, this helped me a lot, actually, during my postdoc. Because at some point, I had to leave Ginés's lab. I tried to resist, but I stayed a few years there, but in the end, I had to leave. So I moved to Herman Steller's lab in Rockefeller University. And at the time, I was interested in tumor biology, because of very, very, very unexpected result. Our research turned from apoptosis to tumors. So I was planning on doing, studying certain aspects of tumor biology. And for that, I wanted to use many tumor models in Drosophila. And since I was working with undead cells, I was planning to use also, as one of the models, these tumor-like phenotypes that we were having with undead cells. But in this case, instead of irradiation, I thought of using the expression of the pro-apoptotic genes RIPER or HIT, along with P35, because this would be a much stronger apoptotic stimuli, and so I was hoping to get stronger phenotypes. And when I did this experiment, what I got was this. So I was still finding the same things that I saw before. I was having undead cells in the posterior compartment, and there was wingless and DPP produced and proliferation in the neighboring cells. But now I was seeing this huge amount of cell death in the neighboring cells, in the anterior compartment. And so this was very weird. And again, the most sensible thing would have been probably to throw this away and continue with my project in tumors. But inspired by Ginés, I guess, I was very intrigued by this phenotype, and I started doing a few experiments here and there to try to figure out what was happening. And in the end, this became my main project. And I found something very exciting, that apoptotic cells not only produce mitogenic signals, they can also produce pro-apoptotic signals that are going to kill the neighboring cells. And the mechanism by which this is happening is that apoptotic cells, at least in this scenario, is that apoptotic cells are producing IgR, which is the TNF homolog in Drosophila, and this is going to activate G and K in the neighboring cells, and so the neighboring cell is going to die. And so, if you do exactly this same experiment, but in an IgR mutant background, or if you block IgR in the apoptotic cells, or if you block G and K in the receiving cells, in any of these cases, you are going to abrogate the cell death of the neighboring cells. So as you can see, this would be a way of propagating apoptosis throughout the tissue, because you can have a primary focus of apoptosis, and through this mechanism, you can spread apoptosis in the tissue. And we thought that this would be an ideal way to kill a large population of cells, because that is happening in several organisms throughout development several times, that you need to get rid of a whole tissue or many cells at the same time in a very synchronized way. And so one of these places is the hair follicle of mice, also of humans. And what happens here is there's a moment where all the cells are going to die by apoptosis in a very coordinated way. And we found that, indeed, in this case also, the apoptotic cells are producing TNF-alpha, and TNF-alpha is important to maintain the synchronicity in the cell death in this scenario. And also you can imagine how this could be relevant to pathologies, because there are many diseases where you have a lot of cell death, and also you have pathologies where the treatment involves a lot of cell death. So in both cases, you can see how the propagation of apoptosis could have an impact on the patients. So basically, as conclusions, first one is that apoptotic cells have many things to say. They are not these silent cells that we're supposed to be. They have many instructions that they can give to the neighbors, and the neighbors in this way will know what to do when they die. Second is that unexpected results have a lot of potential. And maybe it would not be a good idea to write a grant saying that you're going to do random things until you find something weird, and then you will follow that. But actually, I think that unexpected results many times are you know, the way that you find the most exciting things. But most importantly, I hope that you, that you realize how important it was Ginés for me. Not only because he saved my postdoc, because of course the project about the tumors didn't work out in the end, so I was really lucky to follow that weird result, but also because I think that during your PhD, your mind kind of gets wired for science in a way. And you know, later on, the way that you approach science in general, the way you approach the questions that you ask yourself, that pretty much becomes determined during your PhD. And you can see that from the many brilliant scientists that came out from Ginés's lab, and you've heard many during this meeting, they have a certain style. They share many things in common. And I think that many of these things stem from Ginés's talent and Ginés's enthusiasm about science. So I can say myself that I feel very, very lucky and very proud to be part of this Morata school. So thank you very much, Ginés, and thank you all for your attention.